Welcome to the ORS Spine Section Virtual Poster Tour, organized by Drs. Uduruzara, John Martin, and the ORS Spine Section. I am pleased to introduce today's session organizers, Drs. Uduruzara and John Martin. Uduruz is currently working as Associate Professor of Anatomy at University of Health Sciences, Lahore, Pakistan. After finishing her medical school, she moved to the UK and completed her PhD from University of Bristol in 2015. Her postdoctoral fellowship was from University of Hong Kong. Her main research focus is vertebral end plate and its link to intervertebral disc degeneration. She has published over 20 articles to peer-reviewed medical journals, contributed to textbooks, and has made over a dozen presentations at various conferences. She currently serves as co-chair of the ORS Membership Committee of the ORS Spine Section. John received his PhD from University of Pennsylvania in 2015 and is now a postdoctoral researcher in the Department of Orthopedic Surgery at Duke University. His work broadly focuses on connecting spine biomechanics and spine health and is funded by an NIH K-99 Pathway to Independence grant. John currently serves as co-chair of the Membership Committee of the ORS Spine Section. At this time, I am happy to present Dr. Uduruzara.
Thank you for the nice introduction, Sherry. Hello, everyone. On behalf of ORS Spine Section, I welcome you all on our virtual poster tour 2021. The main objective of today's session is to recognize some of the impactful and impressive posters from this year's ORS annual meeting and describe the results to the audiences. Today, we have two speakers who are going to present the posters of their choice. Some of the poster authors have also very kindly joined us. Before I proceed to our first session, I would like to say a few words about our ORS Spine Section that was launched in 2015 at the ORS annual meeting with the aim that the Spine Research Section will play an important role in our diverse ORS community and to the future of patient care. Our mission is to represent the voice of spine research and sciences within the ORS community and beyond, and advance spine research and related sciences to improve patient care through basic, translational, as well as clinical research. To those of you who are not member of ORS Spine Section, I would like you to know that as a section member, you will have many opportunities to engage and connect with the large global spine community. We connect with the spine community through networking, social hours, and our online community forums. We have several section member awards recognizing excellence in research. We cultivate leadership and offer volunteer experiences by giving the opportunity to serve in a section, committee, or as section officers. Our annual section scientific meeting during the ORS annual meeting, virtual scientific sessions, satellite meetings, different other section organized programs are excellent ways of providing education and career development ideas and motivations. At our platform, we have global innovative spine specific teams that include a wide variety of professions and a culture of sharing ideas, time, resources, and results, and much, much more. Here is a list of the awards our section offer. That is Dr. Peter Ruffley Award, Spine Section Travel Fellowship, Annual Section Podium and Poster Awards, that is basically given to the accepted ORS annual meeting abstracts. So join ORS Spine Section to get all these wonderful opportunities. Now with this intro to our Spine Section, let's begin the first session of today's virtual poster tour. Our speaker for this first session would be Dr. Morgan. Dr. Morgan is an assistant professor of bioengineering at Oregon State University. She received her BS in Chemical Engineering from Missouri University of Science and Technology. She's a PhD in Biomedical Engineering from Arizona State University and completed her postdoctoral fellowship at Barrow Neurological Institute. Her interdisciplinary work in drug delivery, transport phenomena, orthopedic surgery, and MRI imaging has been presented in over 24 peer-reviewed journal articles. She serves on the ORS Spine Section Committee as a member at large and also on the Spine Section Membership Committee. I would request Dr. Morgan to please start the session. Thank you.
Thank you, Dr. Zira. Hello, and welcome to our poster tour. Today, I wanted to speak about psychosocial influences on back pain. I found some really great work from this year's annual meeting, and I hope you all enjoy it. I want to start off by offering some context for the posters I'm going to show you today. As a community, we've been aware for a while that back pain is a complex issue. Having visible pathology of the spine does not necessarily indicate that a person will experience back pain. In fact, there are many people walking around with multiple herniated discs and no pain. Here we have a figure from Venti et al., where the X's are painful discs, triangles are non-painful discs, and circles are controls. While there were some differences in the averages, when we look at a lot of these patients in the middle, they have the same disc height ratio and T1 row values. So from these metrics, it wouldn't be possible to tell if a disc is painful or not. The issue becomes even more complex when we discuss chronic pain versus those whose pain resolves after surgery. So let me tell you about Mike. Mike is a back pain patient I spoke to a while back. He is a middle-class white man in his upper 60s, and Mike started having pain when he was 30 years old. He had a discectomy a few years later when he could no longer function. Then he had a fusion in his 40s and then again in his 50s. His pain continued following each of these surgeries, and Mike continues to suffer. He missed many of his children's concerts and soccer games as they were growing up because he couldn't sit or stand comfortably. A surgeon has recommended another fusion because of the continued pain, but there's no specific visible pathology on the MRI, such as a herniation or stenosis. Mike has given up hope and is certain he will bear this pain for the rest of his life. If you ask him, he would say that his back pain ruined his life. So why does Mike, with no obvious pathology, have pain and other people can walk around with hernias and grade 5 discs and have no problem? Well, the spine community has been discussing phenotypes for years now, and this is the data that the idea that there are multiple disease mechanisms that cause disc degeneration and pain, and that we can parse out these different phenotypes to give a more targeted treatment approach. I think today you will see that psychosocial factors very much intersect with this idea of disease phenotypes. It's not just our perception of pain, but also physiological feedback loops to these psychosocial factors that can be seen from the clinic to basic science. Our first poster video, you will hear John Martin discussing clustering and dimensional reduction for visualization of back pain phenotypes. We will also have the first author, Ziyu Wang, joining us today for the Q&A.
My name is John Martin, and the title of my poster is Clustering and Dimensional Reduction, Visualizing Back Pain Phenotypes, Data from the ORS. Back pain is a heterogeneous disease of biological, physical, and psychosocial etiologies. Determining which factors affect an individual, i.e. their pain phenotype, will allow for personalized treatment. Clustering and dimensionality reduction are emerging techniques for visualizing large data sets. The OAI is a multi-center longitudinal cohort study that spans 11 years. It includes 4,800 participants with greater than 1,000 metrics related to their musculoskeletal health. We process this data by identifying subjects with low back pain or greater than or equal to 14 days in the test month. We can remove extraneous variables, including those that were knee-specific or had no gradients, and remove highly correlated variables. We then perform dimensionality reduction by UMAP and unsupervised clustering by KD. Finally, we identified which variables distinguish clusters by statistical analysis. Here's a visualization of back pain subjects in the OAI cohort. Each point represents an individual, and the distance between points represents the relative dissimilarity between individuals. We identified 3 back pain clusters or phenotypes by unsupervised clustering. Cluster 1 had higher socioeconomic status, better mental and physical health, and were primarily white. Cluster 2 had intermediate socioeconomic status, intermediate mental and physical health, and a high calorie diet. And Cluster 3 had lower socioeconomic status, worse mental and physical health, poor nutrition, and were primarily black. At enrollment, Cluster 3 had the most severe back pain, indicated by dark red, and Cluster 1 had the least. This follows the same trends as both socioeconomic status and mental health. Many subjects did not report pain at the 1-year follow-up, as indicated by dark blue. And then by year 10, Cluster 3 still had the most individuals with severe pain. Socioeconomic status, mental health, race, and diet were associated with back pain severity and frequency. There may be an over-reliance on biomedical descriptors of pain that undermines research and social programs that could focus on socioeconomic factors. We're currently using machine learning to develop models that predict which individuals ultimately develop chronic pain. My name is John Martin, and the title of my poster.
Dr. Martin's video highlighted that a patient's mental and physical health correlate with chronic pain. This next video I will show you by Connor Delman talks about an enhanced recovery after spine surgery protocol that includes prehabilitation and early mobilization.
My name is Connor Delman. I'm an orthopedic surgery resident at Epigenetics Medical Center. I'll be presenting on enhanced recovery after spine surgery clinical pathway. Results in a shorter life possible.
Hey, Morgan, it's a little hard to hear this one. Can you turn it up somehow?
There are disclosures. What is recovery after surgery?
Sorry, I can't.
It's defined as a collaborative, multimodal approach to improving surgical outcomes. Goal of URS protocols is to accelerate the recovery process, improve patient outcomes, and reduce overall healthcare costs. A multidisciplinary URS pathway is implemented in single institution. Patients greater than or equal to 18 years old undergoing elective 1 to 3 level lumbar decompression and fusion surgery. Protocol including rehabilitation, preemptive analgesia in the preoperative loading area, preoperative carbohydrate loading, and intraoperative anesthetic pathway, postoperative multimodal pain control, postoperative early mobilization with physical therapy. For study design included an URS pathway that was applied to consecutive patients. The retrospective match score analysis was performed comparing outcome variables pre-URS and post-URS implementation. Looking at a demographic comparison between cohorts, you can see that 57 patients were included in the pre-URS implementation cohort, whereas 56 patients were included in the post-URS implementation cohort. There is no difference in the age of patients, patient sex, or ASA classification. There was a significant reduction in the time to physical therapy after arrival to the PACU. You can see that there was a significant reduction in the time to physical therapy, 21 hours and 13 hours post-URS implementation. There was also a significant improvement in demobilization post-operatively. You can see that there is a significant increase in the number of patients who received physical therapy greater than 20 hours after arrival to the PACU. Most importantly, there is a significant market increase in the number of patients who receive physical therapy within 3 hours of arrival to the PACU, 2% to 25%. There is also a significant reduction in the length of hospital stay, and you can see that the length of stay decreased from 3.5 days pre-URS implementation to 2.9 days post-URS implementation. There is also a reduction in direct patient costs. The average total cost of hospital admission pre-URS was $34,000. Post-URS was $20,848. This was statistically significant. We found that an enhanced recovery after spine surgery pathway can benefit both the patient and healthcare system. Successful implementation of such a protocol requires endorsement and collaboration across multiple disciplines. That concludes my presentation. I'd like to thank you for listening.
I'm sorry that was a little hard to hear. Dr. Delman's work highlighted a cost savings, but consider that it does take a large coordinating effort up front. So hospitals with lower staff and resources might be hesitant to invest the time to get an ERAS program running. This could intersect with Dr. Martin's work showing people with low socioeconomic status have worse long-term outcomes. Next up, we have Ro Garvery talking about exercise attenuation of low back pain in a mouse model. I want to highlight that this work shows molecular and epigenetic evidence for this relationship between pain and exercise.
Today, I'm presenting about the exercise effect on low back pain and epigenetic regulation in intervertebral disc in the mouse model. Intervertebral disc IVD degeneration is one of the major contributors to chronic low back pain. We have previously characterized the spectrum mouse model of low back pain associated with IVD degeneration. Exercise therapy is one of the fundamental treatments for chronic low back pain. Epigenetics refers to several mechanisms, including DNA methylation, that have the ability to change gene expression without inducing any change to the underlying DNA sequence. However, it's still uncertain whether exercise 6 differences were observed there. We investigated the exercise effects on behavioral signs, global DNA methylation, and the messenger RNA expression of genetic regulatory genes in disease. We used 8-month-old male and female spectrum mice and wild-type mice, and they were assigned to exercise procedural groups. Animals in the exercise group received a circular plastic running wheel in their home cage, on which they could run freely. After 6 months of exercise, we performed behavioral tests, global DNA methylation analysis, and the assessment of messenger RNA expression. First, let's see the efficacy of exercise on behavioral signs. You can see speckle mice displayed cold hypersensitivity and actual discomfort. However, exercise significantly improved those signs, especially in speckle mice. The figure on the right shows the assessment of lower DNA methylation. In this figure, we can see exercise decreased DNA methylation in discs, in particular, with the pollen in speckle mice. Finally, let's move on to the messenger RNA expression of epigenetic regulatory genes. These are all DNA methylation-related genes. In this figure, we can see the quite clear difference between males and females. In males, gene expression decreased in the latter genes after exercise. By contrast, messenger RNA expression increased in females in more than half of the genes. This data suggests sex differences in gene expression of epigenetic regulatory genes after exercise. In conclusion, exercise may alleviate lower pain via different epigenetic mechanisms in males and females. Thank you for your attention.
Now let's take a look at a study by Elizabeth Caponia showing that cyclic loading improves the transport of small molecules in the intervertebral disc. As limited nutrition is thought to play a critical role in disc degeneration, we can imagine that cyclic loading from exercise could improve nutrition and the IVD cell's ability to maintain homeostasis.
Hello, I'm Elizabeth Caponia from the Innovative Medical Devices Lab at Rensselaer Polytechnic Institute, and today I'll be discussing a poster titled Cyclic Loading Enhances Small Molecule Transport from the 8-week Degenerated Rabbit Intervertebral Disc. Undergo transamply diffusion to supply the intervertebral disc cells as seen in the left image of Figure 1. Increased biosynthesis causes an increase in cell metabolic activity. The increase in demand for nutrients in combination with an unchanging supply rate results in an imbalance that restricts regeneration and exacerbates degeneration. Illustrated with the right graphic in Figure 1, low-rate loading induces symmetric transport. This transport augments diffusion in the healthy intervertebral disc. The purpose of our study was to characterize the effects of low-rate cyclic loading on small molecule transport in a degenerated disc in vivo. After IARC approval, 49 New Zealand white rabbits were used in the study and were designated to healthy diffusion only, 8-week diffusion only, or 8-week diffusion plus convection treatment groups. Animals underwent a surgical procedure to instrument the lumbar spine. Shown in Figures 2A and 2B, the 2mm pins are placed transversely through the vertebrae to the disc to mitigate their effect on transport. Percutaneous posts are placed dorsally and attached to the pins. This procedure allows for manipulation of a single lumbar motion segment in an awake and non-sedate rabbit. Disc puncture of the 16D channel, see Figure 2C, is performed after post-placement. A bolus injection of danidylamide was given intravenously immediately prior to treatment to simulate transport. For diffusion-only treatment, animals sat quietly. For the diffusion plus convection treatment, animals underwent cyclic loading. Low-rate cyclic loading was applied via a custom loading apparatus shown in the schematic on the right of Figure 3. The left image in Figure 3 shows a rabbit connected to the loading apparatus, the schematic of the attachment in the center. The discs are cyclically loaded to approximately 220 newtons at a frequency of half a cycle per second. Evaluated MRI mapping is used to determine the time constants in 7 regions of the disc, and micro-CT imaging to assess mitochondrial bone density is outlined in Figure 4. For the 8-week degenerated groups, transport peaks at 8.75 minutes for diffusion plus convection and peaked at 12 minutes or later for diffusion only. Additionally, there is a significant difference between 8-week groups of 7.5 minutes and 10 minutes. The data also show that there is a delay in transport for degenerated discs compared to healthy discs. There's no significant difference in subchondral bone density between groups. Differences between the 8-week treatment groups indicate that cyclic loading increases the rate of small molecule transport in a degenerated disc. We can also conclude that degeneration slows the rate of nutrient transport. Importantly, the differences in nutrient transport combined with the lack of differences in bone density implies that changes in transport are due to the extent of degeneration and cyclic loading and not subchondral bone density. These data also suggest that long-term loading can provide a sustained nutrient supply to discs in an enhanced bioactive state. We would like to thank our funding sources and our collaborators that have made this work possible.
The last few posters have focused on exercise, but I wanted to bring our focus back to other psychosocial factors with this last poster. I want to first explain that psychosocial factors have been shown to be integrally tied with immune function. So while the immune system itself is not a psychosocial factor, it is very linked. In fact, this meta-analysis by Shields et al. Showed a number of immune marker decreases in patients undergoing cognitive behavioral therapy, including pro-inflammatory cytokines, immune cell counts, and natural killer cell activity. So the final poster in our session 1 tour today is by Lauren Lishweski, and she discusses immunophenotyping of patients experiencing low back pain.
My name is Lauren Lishweski, and I'm going to be talking about immunophenotyping of whole blood in patients experiencing low back pain. Low back pain is the leading cause of disability and is often associated with disc diseases, which are characterized by a pro-inflammatory environment at the tissue level, with some evidence at the systemic level, but the extent being unknown. For this reason, our objective was to characterize the transcriptome of inflammatory and immune system-related genes in whole blood of patients with painful disc disease, with the hypothesis that patients with low back pain and increased disability exhibit a transcriptome signature characterized by pro-inflammatory activity. Low back pain subjects were recruited from those diagnosed with disc herniation, spinal stenosis, or disc degeneration in the lumbar spine, while control subjects had no history of low back pain or disability. Demographic data was collected from both groups, with ODI data also being collected from low back pain subjects and categorized as either low or high. RNA was extracted from whole blood of all subjects, with the effects of the covariates of gender, age, and BMI being considered, global significance scores calculated, and cell type profiling being conducted. The two main analyses were low back pain versus control subjects and high versus low ODI. First, looking at low back pain versus control, we saw that after controlling for age and BMI, 20 out of 594 genes were differentially expressed, with all of the top 5 pathways being upregulated in low back pain subjects. Then looking at cell type profiling, we saw trends in 3 different cell types. Next, looking at high versus low ODI, we saw that after controlling for gender, 28 out of 594 genes were differentially expressed, with a mix of up and down regulation of the top 5 pathways. A significant decrease was seen in macrophage markers in high ODI, with a trend in 2 other subtypes. Now looking at some example pathways, the upregulation of 20% of the genes in the type 1 interferon signaling pathway supports the increase in T cell marker expression that was seen in the back pain subjects, while the downregulation of the phagocytosis and degradation pathway aligns with the decrease in the expression of macrophage-specific markers in high ODI patients. Finally, in conclusion, age, gender, and BMI are important variables contributing to the whole blood transcriptome of subjects with low back pain. Inflammatory transcriptome and immune cell profiles were altered in low back pain versus control subjects and were responsive to level of disability. These changes suggest an altered immune response, and a fuller knowledge of the transcriptome associated with low back pain would allow for more comprehensive treatment of the associated disc diseases.
So to summarize, we saw that long-term pain was worse in patients who had low socioeconomic status, had poor mental and physical health, poor diet, and were Black. An evidence-based pre- and post-surgical recovery plan, including nutrition and exercise, enhanced recovery. There are epigenetic and nutrient transport changes induced by exercise, and there are immunological differences between people experiencing back pain and disability versus people who are not. Exercise, diet, mental health, socioeconomic status, immune function, and pain all have a long history of complex interactions. There is likely no one intervention that will work for all patients because of this idea of degenerative phenotypes, but also because a holistic approach, including drugs, surgeries, exercise, diet, and psychosocial interventions are all going to be necessary to support the long-term success of back pain treatment. Now I would like to ask some questions of the authors who are here and then open the floor for discussion. I'll start off with Dr. Huang. What types of programs do you envision to help people avoid chronic pain? Diet and exercise programs, more extensive follow-ups, or something else?
So actually, we are thinking using a very innovative way, because as we show in our poster, by using the machine learning algorithm, we can predict the attribution of the patients. So that means we can predict when the first time they visit the doctors, which cluster they're supposed to be, and then we can see how likely this patient is going to develop chronic pain. Actually, this can be done very easily by creating a website based prediction system, which can integrate with the health information system. And then we can tell the patient, you have how many high risk factors that maybe during the future treatment, we can target those factors. For example, this patient is less exercise, or maybe they have mental issue that needs mental health intervention. So we call it like a multi-dimension treatment strategy, which not only targets the pathological factors, but also maybe in the future we can target their mental issues, or maybe more with social economies issues. But that needs not only the doctors, but also the health policy makers to create this kind of like a multi-dimension treatment groups that can better provide better health service to these patients. Yeah.
Wonderful. Thank you, Dr. Huang. Dr. Stone, many of the regulatory genes you studied decreased in male mice who exercised and increased in female mice who exercised, but the pain tests showed the same trends in both genders. Do you have a hypothesis for why that would be?
No. If anyone else has a hypothesis, that would be awesome. It suggests that even though they have similar pain conditions and they respond similarly to the treatment, the mechanism driving the therapeutic benefit is different.
I think that's really interesting. Thank you, Dr. Stone. Ms. Leszewski, your study looked at subjects with low back pain and pathology versus no low back pain. Were you able to distinguish if there was asymptomatic pathology in the control subjects? And what would you hypothesize if you did have a painful pathological group versus an asymptomatic pathological group?
So we were not able to determine the asymptomatic pathology because that would require MRI imaging of the control subjects, and we didn't perform that. But we were able to look at the clustering of the pathway scoring and see if any of those control subjects fell within the cluster of patient subjects. And I would guess that those would probably be the control subjects that were closer to showing some sign of pathology. And then in terms of an asymptomatic versus a painful pathology, I would predict that the painful pathology would show greater expression of pain and inflammatory and immune-related genes, while asymptomatic pathology would show less, and then control with no pathology would show less than that.
Thank you, Ms. Leszewski. Please tell me when my time is up, but I'll start going through some of the audience questions in the chat, or I don't know if Aruj is going to be asking those, but for Lauren from Simon Tang, great work. Do you think the immune response is triggered by the low back pain, or that the immune response is a pre-existing condition that is amplified by the low back pain? How might you decouple these?
So I think that the immune response is actually a pre-existing condition. I've read some literature that looked at acute pain versus chronic pain, and then acute pain that has transitioned into chronic pain, and they actually saw that there was similarities between the acute transitioning to chronic and the chronic pain. So I think people are generally predisposed to developing this chronic pain.
Thank you. We have a question from Dr. Muhammad. How can we actually relate mental health with back pain issues? And can you tell in which mental health conditions there is probability of back pain issues? Laura Stone says lots of data shows links to both anxiety and depression. I would agree with that. There are mental health surveys you can take prior to surgery that are predictive of chronic pain following surgery. And I think there is quite a bit of a feedback loop involved. If you think about people who suffer from long-term pain, that can be a very depressing situation, right? So I think it works both ways. People who are experiencing pain become depressed, and then you get, all of these complex factors. The immune system gets involved, and your perception of pain is different. So it's just this whole feedback loop, in my opinion, that kind of, you have to try to address in multiple different ways to try and break those patients out of that loop.
All right, I think we're going to have to thank you, Morgan. That was great. We're going to have to move on to Dr. Garvery's presentation now. And also thank you to all of the poster authors who came to help us out here. So that was really great. So I'm going to introduce the second speaker here. This is Dr. Ro Garvery from McGill University. Dr. Garvery is an assistant professor with the Division of Orthopedic Surgery in the Department of Surgery at McGill. His research interests lie in intervertebral disc organ culture models and developing innate small molecular therapeutics for the treatment of disc degeneration. He has been closely involved with the spine research community for many years and serves on the membership committee currently of the spine section within the ORS, as well as various other outreach and collaborative initiatives at his own institute. So we thank Dr. Garvery for joining us today, and he can take it away here.
Thanks a lot, John, for the introduction. Hello, everyone, and welcome to the second session of our poster tour. The thematic of this section is intervertebral disc therapeutics, from small molecular targets to injectable hydrogels. And this is my disclosure. So I want to start off by saying that all the presented spine and IVD related posters and podiums at ORS annual meeting 2021 were of very high quality and caliber. And this selection of posters in no way demerit the ones that were not selected or included in the tour. I will briefly tell you about the choice of posters and the flow. Just for the introduction, novel therapeutic strategies are being developed for intervertebral disc regeneration and restoration of function. Current focus is to develop non-surgical therapies and advance the field of IVD, tissue engineering, and regenerative medicine. These therapeutics range from small molecular signaling pathway targets, bioactive compounds, and peptides to innovative biomaterials for structural restoration and drug delivery with or without cell-based therapy. This recent paper very nicely summarized that what is the need of the hour, that we have biomaterials, we have biomolecules, and we have the cell-based therapies, and how to amalgamate them for proper delivery. Because sometimes we have seen that we have a proper therapeutic agent, but the desired effect is not there. So for the choice of posters, you can see that this is the therapeutic strategies that these studies adopted. So the first three posters are chosen for their exploration of potential small molecular therapeutic targets. The first two present the role of calcium signaling pathways through calmodulin 1D, transmembrane protein 38B, PTYH2, intercellular calcium ion, TRPV4, and increased calcium fluxes. The third presentation evaluates the role of rapamycin on the age-related rescue of IVD degeneration. Rapamycin and its downstream targets are known inhibitors of mTOR, which is a negative regulator of autophagy. Calcium ion is the most highly and most tightly regulated ion in the body, and is also related to mitophagy and sending a death signal to the nucleus via mitochondrial pathways. The next two presentations after that show or present the work based on hydrogels in caprine models of IVD regeneration, one in vivo and other in vitro organ culture models. One of the hydrogels acted as a standalone restorer of disc height, and the other had cells embedded in it. Together, these selected posters present the spectra of development of therapeutics for IVD regeneration, ranging from potential intracellular signaling targets to biomaterials that can be used to deliver inhibitors or to promote these signaling molecules, and cell-based therapies or act as a standalone therapeutic agent. In this session, you will also see the use of various experimental platforms being used for IVD research, from 2D cell cultures to in vitro whole organ culture models, both small and large, and in vivo preclinical large animal and small animal models. In our first poster, you will hear Mr. Efetse discussing the role of verteporfin on restoration of human NP cell shape and the role of calmiodulin 1D on the NP cell physiology.
I am Eri Afetse, and today I'll be presenting on how verteporfin induces changes in NP cell shape, but also induces the differential expression of calcium calmodulin protein kinase KMK1D. NP cell shape changes with age from a more rounded shape to a flattened-like shape. A number of signaling pathways regulate NP cells morphology, one of which might involve a calmodulin-dependent kinase. Calmodulin-dependent protein kinases are important for cell function and influence blood processes like gene transcription and cytoskeletal reorganization. Our hypothesis is that verteporfin will induce cell rounding with decreased KMK1D expression, and siRNA-mediated knockdown of KMK1D will promote healthy NP markers expression. Two experimental methods were used to test our hypothesis. For the first one, primary human NP cells were obtained from surgical waste and treated with DMSO and verteporfin. RNA sequencing was performed, and the cells were also stained for F-actin fibers. For the second one, KMK1D knockdown was achieved with siRNA transfection, and gene expression was quantified with qPCR. The result of the first experiment suggests prominent F-actin fibers in cell treated with DMSO, figure A, and reduced F-actin fiber in cell treated with verteporfin, figure B. Figure C shows a highly aligned actin in cell without verteporfin, and figure D shows decreased expression of KMK1D over the 4 days period. The result of the second experiment showed a transfection efficiency of 94% and increased NP cell phenotype marker in knockdown KMK1D cells. IPA was used to analyze RNA sequencing data to determine a role of KMK1D and figure out its involvement in biological pathway regulating NP cell morphologies. In the pathway presented here, most of the genes are shown to be downregulated, and it shows that KMK1D is involved in cytoskeletal rearrangement process. KMK1D has been linked to glucose regulation, changes in the cytoskeleton and fibroblastic characteristics. Results from this study contribute to the goal of understanding how changes in NP cell shape play a role in maintaining a juvenile-like NP. These are my acknowledgement and references. Thank you.
Mr. Feste's video highlighted the role of cell shape that can be interplayed with NP cell physiology and KMK1D as a potential gene target for IVD regeneration. Also, the role of TTYH2 and TMEM38B is also very interesting and needs further exploration. The next video I will show is presented by Mr. Eason, and he talks about the role of TRPV4, calcium channel agonist GSK101, and its role in activating this channel and causing increased intracellular calcium fluxes and a dose-dependent increase in GAG accumulation in a mouse tail whole organ culture model. We also have the senior author, Dr. Simon Tang, joined us today for the Q&A session.
Hi, I'm Garrett, and today I'll be talking about how the activation of TRPV4 mediates calcium signaling and increases GAG production in the invertebral disc. It's well established that dynamic physiological loading induces an anabolic response in IBD. This anabolic response consists of an upregulation of factors such as GAG, COL1, and COL2. However, it's not well known how this initial dynamic loading is transduced into this anabolic response. In chondrocytes, it's been shown that TRPV4, an ion channel, is responsible for mediating mechanotransduction, and following dynamic loading is responsible for promoting an anabolic response in these chondrocytes. As chondrocytes are similar to IBD cells, it's been proposed that TRPV4 activation may also mediate IBD anabolic response to loading. First, we looked at whether or not the activation of TRPV4 could induce calcium signaling in IBD. We cultured whole organ lumbar IBDs and cultured them in infrared and fluorophore calcium indicators. We then used confocal microscopy, and adding GSK101, a TRPV4 chemical activator, we observed whether or not the activation of TRPV4 does indeed initiate calcium signaling in the IBD. In the video on the right, red cells have low intracellular calcium and the green cells have high intracellular calcium. As you can see, as we add GSK101, our TRPV4 activator, we see this high increase in high intracellular calcium signaling, indicating to us that GSK101 and the activation of TRPV4 does in fact initiate calcium signaling in IBD cells. Following this, we performed a GSK101 dosing experiment in which we activated TRPV4 for 7 consecutive days at 3 different concentrations. At the end of this experiment, we then performed DMFV and contrast-enhanced micro-CT to measure GAG production. On the left, you can see that as we increase our GSK101 dosing concentration, we see a significant increase in overall GAG production in our IBD. This can be seen visually through contrast-enhanced micro-CT. What you can appreciate on the right is an increase in overall attenuation in the nucleus pulposus of our TRPV4 activated IBDs, showing a higher GAG content in the NP in our GSK101 dose discs. So to summarize, the activation of TRPV4 by GSK101 induces calcium signaling in IBD. TRPV4 activation increases GAG content in IBD, particularly in the nucleus pulposus. And this establishes the complex role of TRPV4 in IBD and the need to further investigate its downstream pathways. And with that, I would like to thank members of my lab as well as my funding sources.
Mr. Eason's work highlighted the role of mechanotransduction and increased calcium fluxes, causing the triggering in anabolic state of the mouse IBD in organ culture. This intersects with Mr. Efetz's work, as increased calcium can also trigger calmodulin K and its phosphorylation and the downstream signaling pattern. Calmodulin K is a very diverse family and needs to be explored, that which CAM-K is upregulated with regular mechanical and drug stimulation, and what is the optimal dose or loading regime to trigger these changes. So next up, we have Ms. Crystal talking about the role of rapamycin administration on intravertebral disc degeneration in a naturally occurring model of aging and the rescue of IBD degeneration in a non-human primate marmoset.
Rapamycin has been shown in several animal models to increase lifespan and decrease aging and age-associated pathologies. The question is, how does rapamycin influence age-associated IBD? And the goal of this study was to determine the effect of rapamycin treatment on age-associated IBD in marmosets. Male marmosets were used for this study. Marmosets were aged to young or old time points. A subset of the old marmosets were given treatment of rapamycin via oral feeding of yogurt plus the rapamycin capsules. This treatment was given daily for 1 year prior to sacrifice. All marmoset spines were harvested for analysis in our lab. The outcome measures assessed were disc histological features, matrix anabolism, matrix catabolism, and autophagy markers. Figure 1A shows histology photos of sections stained with H&E from 3 animals in each group. NP and AF photos are shown. Legend indicating the meaning of the errors is to the right. Figure 1B shows the quantified composite histological score. Older marmosets had a significantly higher histological score compared to young marmosets, indicating significant degeneration had occurred in the disc. Rapamycin treatment given to old marmosets significantly decreased the histological score. Figure 1C shows a detailed histological scoring that was used to generate the highlighted composite score. Disc matrix anabolism was shown both quantitatively in A and qualitatively in B to be significantly increased in old marmosets given rapamycin treatment compared to normally aged marmosets. Disc matrix metabolism in young compared to old marmosets was shown in C to be significantly increased in old marmosets, but in D, no significant effect was seen in old marmosets given rapamycin compared to old controls. Disc autophagy markers in E showed potentially impaired disc autophagy in old marmosets, which was restored in those given rapamycin, based on LC32 and P62 data. Other disc autophagy markers show no clear trend of increased or decreased autophagy in rapamycin-treated marmosets. Rapamycin treatment significantly restored proteoglycan homeostasis and significantly improved histological score in the disc of aged marmosets. This implies 1 year daily rapamycin treatment delayed IDD progression. Disc autophagy-related proteins were not significantly increased with rapamycin treatment. Other studies show rapamycin treatment in other tissues does significantly increase autophagy. This suggests either rapamycin given orally cannot diffuse into the disc or autophagy is not regulated by rapamycin in the disc. This is a novel study of the in vivo effects of rapamycin treatment on age-associated IBD in marmosets, and some beneficial outcomes on disc aging were reported. This is the first study to use a non-human primate model to study age-associated IBD in rapamycin treatment. Future studies are warranted to confirm findings and investigate the therapeutic effect of rapamycin on age-associated IBD.
Ms. Christian's work showed that the degenerative process in the IVD can be rescued by rapamycin administration, and the histological scores and metabolic activity of the IVD in treated old marmosets were better than compared to untreated old marmosets. The study, however, could not show autophagy, which they attributed to the failure of drug delivery as a potential cause, wherein the bimetallic drug delivery system for this particular model can play a very major role. Now we scale up our vision, and we will watch our next presentation by Dr. Fusco, where she presents her work in vivo K-prime model of enzymatically induced IVD degeneration, wherein a non-cross-linked hydrogel was injected as a standalone therapeutic agent without seeded cells. This poster shows the therapeutic outcomes in a long-term survival study and the use of large animal models as preclinical models of IVD research.
Hello, I would like to thank ORS for this opportunity, and I'm here today to talk about the research title Hydrogen Neuroplasty improves disc height and cognition in a goat model of moderate severity disc degeneration. Low back pain is a leading contributor to disease burden worldwide with high societal and economic costs. Intervertebral disc degeneration is strongly implicated as a cause of low back pain, with early degenerative changes occurring with nucleus pulposus. Our study objectives were the development of a non-cross-linked nucleus replacement hydrogel, the development of percutaneous hydrogel delivery method, and finally the evaluation of short and long-term potential of this hydrogel in a clinically relevant large animal model. This presentation will focus particularly on this last aspect. For our study, we used 6 skeletal immature goats and there were 2 percutaneous surgical procedures, one for the delivery of CABC at 3 intervertebral lumbar levels to induce disc degeneration, and 2 weeks later, neuroplasty by hydrogel delivery at 2 lumbar levels. 2 animals were sacrificed at 6 weeks and 1 animal at 12. 3 animals were enrolled in a long-term survival cohort for which evaluations are still ongoing. Antemortem evaluations include clinical observations, radiograph CT, and disc height measurements. Postmortem evaluations include micro-CT, explant analysis, and histological grading. Clinical observations showed an eventful recovery in conventional survival times. Polymer exclusion in the spinal canal occurred in 2 animals after rupture of the annulus fibrosus during hydrogel delivery, anyway without major clinical repercussions. The long-term survival cohort animals appeared clinically normal up to 2 years after surgery, as you can see in the video below. Radical sacrificial stability of the implant up to 18 months after surgery. After 2 weeks of CAVC from CAVC injection, this guide decreased up to 80% of pre-CAVC levels, and then improved after nucleoplasty in the treated discs, while the degeneration continued in the untreated discs. The micro-CT showed increased cortical and trabecular bone volume fraction at treated levels between the 12 and 6-week survival cohorts. The extant polymer analysis revealed higher elastic moduli and polymer content compared to non-implanted polymers. The histological grades were overall improved compared to untreated discs. In conclusion, the results of this study support the short and long-term efficacy of the injectable hydrogel in normalizing the mechanical function in the degenerative disc. The implant did show extrusion following delivery, and also orthotic changes in the treated and distant levels were observed even in longer-term follow-up radiographs. Therefore, hydrogenucleoplasty may represent a minimally invasive therapeutic solution that preserves joint mobility, potentially mitigating a decent level of disc degeneration. With this, I conclude my presentation. I would like to thank you for your attention and thank PGFTech for funding this study.
From this study, we move on to the last presentation of this session, where Dr. Snug talks about the in vitro caprine IVD organ culture model for testing Leponite cross-linked P-NEPAM co-DMAC injectable hydrogel, also known as NVGel. This talk covers the last color of our session spectra, which encompasses the use of large animal whole organ model for IVD, which has been scaled up to match human IVD dimensions. We also have Dr. Lemaitre here with us for the Q&A session.
Hello, I'm Joseph Snooks, a postdoctoral research associate at Sheffield Hallam University, UK. Here I'm presenting a Caprine model of intervertebral disc degeneration, a testing platform for an injectable hydrogel. There are many different animal models of IVD degeneration, yet not all accurately represent human degeneration. Here we aim to develop an ex vivo organ culture model that mimics a moderate level of human IVD degeneration to test the efficacy of emerging treatment strategies such as Epigel, an injectable hydrogel showing promising anti-degenerative properties. The model was produced by using a loaded disc culture system in Figure 2 that allows the culture of intact IVDs under loading and media perfusion. Figure 3 shows that Caprine IVDs were preloaded for 3 days and injected with a combination of collagenase and chondroitinase ABC and subjected to 7 days of simulated physiological loading. To test Epigel, this regime was repeated, and then Epigel alone, or with mesenchymal stem cells, was injected and discs were cultured for a further 21 days. Figure 4, during culture, the height of controlling sham discs remained constant, whereas enzyme-injected discs in red displayed a reduction in height over time. Mass and trichrome staining in Figure 5 shows the presence of fissures and matrix loss in enzyme-injected IVDs. IHC Figure 6 highlights a decrease in matrix protein expression and an increase in catabolic protein expression in enzyme-injected discs. Moving on to NP gel injection, Figure 7 displays macroscopic images of Caprine IVDs where injected NP gel is clearly visible within NP regions. Below, in Figure 8, mass and trichrome staining shows the integration of and NP cell migration into NP gel within discs. Immunohistochemistry in Figure 9 shows an increase in matrix expression and decrease in catabolic proteins in discs injected with Epigel compared to degenerate controls. In conclusion, we have established a relatively quick, reproducible ex vivo Caprine model mimicking human degeneration. Injection of Epigel attenuated the catabolic phenotype, suggesting it has a potential to halt degenerative processes. Finally, this model may have the capacity to enable rapid screening of novel strategies to combine IVD degeneration in the future. Thank you.
To summarize, many advances have been made in the field of IVD research with identification of numerous drug targets, therapeutic agents, modification of mechanical stimuli, cell-based therapies, and the use of biomaterials as standalone bioscaffold or as a therapeutic delivery agent. These advances need to intermingle, as when we deliver a potential drug target or therapeutic agent, we also need an optimal delivery system. Thereon, we need a preclinical testing platform, either in the form of organ culture models, both large and small, and large and small animal models of naturally occurring and induced disease, and the evaluation in both quadrupeds and bipeds, non-human primates. With this session, we wanted to cover the exciting research being done in the field of IVD physiology and pathophysiology in a breadth as wide as possible. I hope you enjoyed the session as much as we enjoyed hosting them. Now I would like to ask some questions to the authors who are here and then open the floor for questions and discussions. Thank you.
Can I ask a question?
Yes, Laura, please.
This might be really naive, but so if just injecting hydrogel is therapeutically beneficial, does that raise problems for all of the 3D models that are embedded in hydrogel?
That is a very good question, and the author is not here to answer the question. That was another question that I had in mind, because the same group is using another hydrogel, which is thermal crossling gel, and is putting cells in there. Same as Dr. Lemaitre's group is also augmenting these hydrogels with cells. So in my opinion, if you have a hydrogel that is there, it might lead to a failure. So I would, in my personal opinion, would like to have something augmented so that when the cell breaks down or the gel breaks down, there is enough cellular machinery that has taken over in the meantime and has stimulated the matrix production, rather than relying solely on a gel. Because that's where we are just relying on, say, the nuclear implants that we have for surgical interventions, which might have failed. So which kind of is counterintuitive. Dr. Lemaitre, do you have any thoughts on that?
Yeah, so I think, that's a really interesting question, Laura, in terms of the kind of 3D cultures as well as cells in the lab and looking at how the cells behave, because a lot of the hydrogel systems that we're looking at for regenerative purposes do have these biological effects on the cells. Now, I think when you're looking at 3D culture of cells and look more at how those cells behave, you need to use much more inert, if you like, hydrogel systems. So alginate's a common one that's utilised, and alginate is used to maintain phenotype of the cells, but doesn't induce an effect on sort of anti-catabolism and things like that as well. But certainly if we do studies with the hydrogel system that was described by Joseph, if we do treatments with cells in that hydrogel with things like interleukin-1, we actually inhibit the effects of interleukin-1 when we culture them in that hydrogel as opposed to if you culture them in alginate. So you do get different responses if you culture in different hydrogel systems.
So I'm looking at the chat room right now, and there is a question for Dr. Tang from Dr. Gears. Could you inhibit during the day and activate at the night? The whole TRPV administration schedule.
Yeah, I think that would be certainly possible, because the half-lives of the agonists and antagonists are I think right on the order of 12 hours. So it'd be possible to kind of do, come up with a dosing regimen where it's inhibited. And I think what you're getting at is kind of like that diurnal response, right? So maybe like promote growth or stimulate loading maybe during the day, and then maybe when you're doing activities actually inhibit loading so you prevent damaging loads. Is that kind of what you're thinking? Yeah, that's a really good idea.
So Dr. Tang, in follow-up to that question, I have another question for you. Will it be possible to have a depot or a sustained release prep implanted either in bone or subcutaneously that continuously releases? Or is it imperative to have it locally administered, not systematically? Because there are calcium channels all throughout the body.
So I would imagine, certainly to achieve that like diurnal, response, like a local sustained release would be a really, really great idea. I think what we need to be mindful of is the wide effects of TRPV4 on neurons. So it is highly expressed in neurons. So we want to make sure we're hitting the things we want and not the things we don't want. So and I think there's, one of the things we're looking at is kind of the interactions between the neurons and the disc, and whether TRPV4 is a conduit for that response.
So Dr. Tang, last question for you was on my list. So with the GSK 101, have you seen any muscle twitching or muscle flaccidity because of the calcium channel, calcium ion interference? Because they also modulate calcium a lot. These are the neurotransmitters.
That's a really good question. We haven't specifically looked at it. So, a lot of this work is just done in organ culture. So we have the luxury of not having the muscle confound our readings. I think Dr. Gilak's lab has done kind of the performance and behavioral evaluations on these mice, and I don't think he has seen any behavioral differences following like an injection. I don't think he specifically looked for muscle twitching. So, I think one of the things that we do know is that TRPV4 is most responsive to sort of like lower frequency loading. So there's a whole host of other like ion channels that work in concert to respond to different like mechanical loads, both in magnitude and in frequency.
Thank you. I have a question for.
I think, unfortunately, sorry, Ro, we're running over time here, so I think we have to cut it off here. But I want to say thank you to all the speakers, and also to Sherry from ORS for helping us put this all together, and for all the poster authors who came here. So thanks so much, everyone, and have a great rest of your day.